Good evening, everyone. I'm Akilandeshwari. I'm from IIT Jodhpur. I'm currently doing my master's. So, the title of my project was Rapid Detection of Inflammatory Biomarkers using Surface Enhanced Raman Spectroscopy. So, let's make it easier for the audience here. I would like to split the title into simpler terms and make you understand. So, first, I would like to talk about what are inflammatory biomarkers. Inflammatory biomarkers are nothing but proteins which are basically released during inflammation. Inflammation is the response of the body, human body, to injury or infection. So basically, when an inflammatory cell is actually having exposure to an infection or an injury, what happens is that it gets into an activated stage and later it releases inflammatory biomarkers, which are central to a lot of diseases. So, to name a few of them, I've mentioned COVID 19, chronic cancer, and also autoimmune disorders. Later, now talking about the principle of Raman scattering, it is basically the structure and chemical composition of every material or um, every material can be done by using the vibration of molecules which are specific to the functional groups present. So, just to enhance the reading of the Raman scattering, what we are doing is that we are using surface enhanced Raman spectroscopy, which is which is to amplify the Raman signal intensity by using metallic nanostructures or nanoparticles. We are using label-free source detection technique in our study. Now, talking about the workflow, firstly, I had to select key pro-inflammatory biomarkers, which are interleukin 6, interleukin 1 beta, and TNF alpha. These are the three prominent pro-inflammatory biomarkers which are generally released during inflammation. During an onset of a disease, the few of the most important biomarkers which are released are these three which I have mentioned. So later I had to do a literature survey or literature study to understand what are the characteristic Raman peaks for each of them. The third step was to detect the individual biomarkers on the source substrate which was prepared by Mr. Sarvar Singh and later perform the simultaneous detection of the biomarkers in a mixture, which is basically detection of all three of them together on a source substrate. And the last step was to detect the biomarkers in various biological liquids, samples like urine, saliva, and serum. The last two steps are actually the work in progress. I've just added a visual representation of what was the workflow that I did. For the literature review, every crystal structure of inflammatory biomarker has specific peaks which are observed in literature. So my aim was to first detect the individual biomarker sample by using source substrate. So the incident laser is shined upon the sample and what is the scattered light will be analyzed by Raman and basically it can compare we can compare that with the Raman signal that we get in the literature and conclude what are the cytokines present. Now the second part was to detect the source by using a mixture of biomarker sample and the last one was to use serum samples. Now coming to the literature review, what I did was the first part was to understand the existing methods which are generally used for the detection of inflammatory biomarkers. So to start off with, there are a lot of techniques which are bead-based assay techniques and ELISA techniques. The major drawback that we all face in this technique is that it has a long antibody incubation timing. So what happens is that when you're giving a sample to any unit or a clinic, it almost takes about 1 or 2 days to get the report. So basically what we are doing in SIRS label-free detection is to reduce the timing which can be used for detecting the cytokines. Now for the second part is to understand the characteristic Raman peaks from the literature and to correspond every bond present in that molecule to a particular peak in the Raman signal. And the third one was to do a study based on what will be the maximum detectable limit of an inflammatory biomarker in different samples, be it serum sample, urine sample, or saliva. Now, this is what I was explaining about. So, in Raman spectrum, the example that I've mentioned here is for interleukin 6. So, every peak that have been mentioned here in the literature has corresponding band assignments, just like Sarva sir mentioned. Now, talking about the materials and methods, I had used the biomarker stock solutions, which we generally get it in the ELISA kits, and the concentrations of them are mentioned here. Later, I had to deposit them on the SIRS substrate individually, and I had to shine upon the laser to detect the inflammatory biomarker. The major task here was to standardize the amount of laser power intensity and integration time which has to be used on the sample to detect the biomarker. I had to play around with the intensity, the power intensity, and as well as the integration time to find out what can be the optimum amount of information given to detect the molecule. Now, the second part of the materials and method was to use two different wavelengths of light because I'm trying to standardize my technique. I had to use two different wavelengths. When I used 532 nanometer, I could observe that the sample was getting degraded because, as we all know, that 532 nanometer has a higher laser energy compared to the 785. So when I use 785, it could prevent the degradation of the biological samples. Later, I had to standardize based on whether it can be wet samples or dry samples. The reason I did this was because in literature it was mentioned that the dry samples had more tendency to bind to the source substrate rather than the wet condition. So I had to do experiments to compare and find out which one is better. The last one was to enhance the Raman spectrum peaks that I get. For this, I had to mix the inflammatory biomarkers with gold nanorods. So this basically creates a three-dimensional mesh-like structure, which is like a sandwich structure. The first one will be the silver nanoparticles, which are already there on the source substrate. Later are analyte, and over that we are trying to add gold nanorods, which can even more enhance the Raman signal. Now coming to the results part, as I mentioned, I started off with doing wet samples of interleukin 6. So I have mentioned the corresponding peaks intensity just to show the comparison. To start off with, the figure 1 shows interleukin 6 sample, which is standard solution from the ELISA kit on the source substrate. So these are the two peaks that were observed. When we observed the same thing using interleukin 6 and gold nanorod sample, I could observe that the intensity of the peaks has more or less been same. Like what I'm trying to say is that the wet samples couldn't really show me good results. But when we come to the dry samples part, for the drying step, what I did was after dropping the analyte on the source substrate, I had to dry it in an incubator for about 20 minutes. And later, I took the reading. This is for only interleukin 6. Similarly, this is for interleukin 6 and gold nanorods. I could see that the corresponding peaks that I've mentioned here, there was a drastic increase in the Raman signal peaks because of the use of the gold nanorods, which also increased the specificity of what I'm targeting. Now talking about the second part, which was interleukin 1 beta. More than talking about characteristic peaks, we have to also focus on something called characteristic patterns of the inflammatory biomarkers which can be observed. So here, the pattern what I'm talking about is this peaks that I'm mentioning. They are all following a particular trend. So this trend has to repeat in all my experiments. So I should show that they are replicative so that I can conclude that these are the characteristic peaks. Now talking about the final interleukin which I used was TNF alpha. So the peak here has lower intensity, but here when you see there's a steep curve. I could find the steep curve between 1415 to 1782 in every replicative experiment that I did. Now coming to the results part, this is basically summarizing all the characteristic peaks that I've mentioned. So interleukin 6 had these many number of characteristic Raman peaks, the shifts, and interleukin 1 beta and in TNF alpha. The main aim of this experiment was to standardize what can be the optimum power intensity and laser timing that can be used to detect these inflammatory biomarkers on the source substrate. So these are the peaks which are specific to the interleukins that I've mentioned here. Now coming to the conclusion part, I have shown you here the representative data of the characteristic peaks of all the inflammatory biomarkers. I had to play around with the inflammatory biomarkers. I had to start. I have to first conclude what are the inflammatory biomarkers to be used, and later I had to work on what can be the different biomarkers and their relation to different diseases. And my work in progress now is to detect simultaneously all the inflammatory biomarkers together in a mixture. For now, what I did was to individually detect them, and later my step, which I'm doing right now, is to simultaneously detect the Raman signal from the inflammatory biomarkers. And the future work or the long term goal would be to simultaneously detect the inflammatory biomarkers in biological samples, especially in time sensitive settings, because that is our main concern. And the last part is to study on the detectable limit of inflammatory biomarkers on the search substrate. Detectable limit, what I'm trying to say here is that I'll be drawing a concentration curve of different cytokine solutions that I'm using, and as well as I can compare that with the search substrate readings and to conclude what can be the detectable limit. Um, I would like to acknowledge IIT Jodhpur for funding my research and the Center for Research and Development for providing the infrastructural facilities for my work. And I would like to thank Dr. Ajay sir and Mr. Sarvar Singh for their constant support and guidance. Thank you. That's all from my side. I'm open to